Hey everyone, welcome to Professor Long's Lectures in Anatomy and Physiology. I'm Professor Bob Long. These videos that you're watching are intended for use by students enrolled in my Human Anatomy and Physiology class. If anyone else finds them helpful, by all means use them. Now we've been talking about blood. We've done 4 or 5 videos on blood. They're all going to be loaded on my YouTube page. This last video is over blood typing and how does it work. We use what's called the ABO typing system. Now the way ABO typing works is it works basically on the same kind of concept as the immune system with antibodies and antigens. So before I get into it, let me give you the setup. If I take a red blood cell, I don't have a red marker on me, so let me grab a few, just in case one of them is not very good. If I take a red blood cell, this red blood cell is going to have proteins on its surface called surface antigens. Some of them have certain shapes to them. Some of these proteins are normal proteins that are on many of our cells, but a couple of these proteins might be unique to your red blood cells. So we're going to look at these cell surface antigens. Now before I go into too much detail, there's a concept called a linked gene. So let me go into a different thing first, and then all of this should make sense. If I take a typical human cell and I look inside the nucleus, there's all these pieces of DNA called chromosomes. And along each chromosome, there are sections called genes. Each gene is going to be the instructions on how to assemble one protein. Now, if I have, let's say, these 6 genes here, if I need to make a protein, there's a way to go in and turn on a gene, and when I turn that gene on, it's going to give me a protein. That protein can stay in the cytoplasm of the cell, it can get embedded in the cell membrane, or it can be secreted. But that's how we know when to make proteins, is some signal turns a gene on, or it can turn a gene off, and you stop making that protein. It's kind of how hormones work. When a hormone binds to a receptor, that can kick off an enzyme cascade that will turn a gene on or turn a gene off in the target cell. Got it? Good. Now, some of those proteins are made every day by our cells, like cell surface markers. Now, sometimes if I turn on one gene, let's say I'm going to go in and turn on this gene also to make a new protein. That gene might be linked to another gene, so that if I turn this one on, I automatically get this one, whether I like it or not. Everybody has sort of a hallway or some kind of, you've been in a house where there's a hallway that has a light switch at either end. If one light switch is up and the other one's up, the lights are on. If I flip one down, the lights are off. If they're both down, the lights go on. If one's up, one. So they're linked. They both affect the lights. If I turn this gene on, I get that gene. They call those linked genes. Now, when it comes to making some of these antigens on the surface of our red blood cells, that gene can be linked to a gene for a specific antibody in white blood cells. So if I turn on this gene in one cell, it turns on a gene in another cell, and I'm going to get 2 separate proteins. It's kind of how this works. Now, when we do the blood typing, okay, we're going to talk about linked genes. Some of these genes are linked and some of them are not. I'll explain what I mean. So we're going to do what's called the ABO typing system. Now it's much more complex and there's been more figured out, and nowadays you put blood in a machine and press a button and it spits information out. This is how it used to be done. Okay. So there's going to be different blood types. When I list the blood types across the top here, you can have type A, you can have a person that has type B blood, type AB blood, or type O blood. Okay? I left a lot of space there. Let me rearrange this so I have a little bit of room to do the positive and negative. So for the blood types, we have type A, type B, type AB, and type O. We're also going to talk about positive and negative blood, but that's a whole different topic, so to speak. Now, when it comes to your blood type, you need to know this. You need to know which surface antigen is on the red blood cells. You also need to know which antibody is produced by this person's lymphocytes. These genes are going to be linked very often. If I have a certain blood type, I'm going to get a specific antibody in my plasma. If I have a different antigen, I'm going to have a different protein in my plasma. So you need to know which antibody is produced by lymphocytes or which antibody is in your plasma. So now, if I have type A blood, my red blood cell is going to have little proteins on the surface that we're going to designate with the symbol A. They really don't look like that shape. It turns out that your lymphocytes will automatically make antibodies that have the B or that are going to be against a B surface antigen. So you have anti-B antibodies in your plasma, and you have the A surface antigen. So anyone with type A blood is going to have A surface antigens on their red blood cells, and since that gene is linked to this gene, they're going to have anti-B antibodies. Automatically, you're born that way. Now, someone with type B blood, if I were to look at their red blood cells are going to have little B surface antigens. As a matter of fact, this tells you which surface antigen that you're going to have. A person with type B blood is going to have these antibodies in their plasma. They're going to have what we call anti-A antibodies. And they're going to have the B surface antigen, which is where I like to ask my questions. A person with type A blood would have which of the following? They would have A surface antigens, anti-B antibodies. A person with type B blood will have B surface antigens and anti-A antibodies. Now, if somebody has AB blood, then their red blood cells are going to have both the A's and B surface antigens. Sorry, I dropped that. So what we can say is that this person is going to have both A and B surface antigens. They don't make either antibody. We can say that they make neither anti-A nor anti-B antibodies. Okay? We don't say they make any antibodies, they just don't make these two antibodies. Now, I need to make a little bit more room, so I'm going to erase a little bit of this. Sorry. If you have type O blood, then this person's red blood cell is going to look like this. It's not going to have the A, and it's not going to have the B antibody. So we can say that this person has neither A nor B surface antigen. They don't make either one. And in their plasma, they are automatically going to have this. They're going to have both the anti-A and the anti-B antibodies. I'm not going to write all that out, but you could say they have both the anti-A antibodies and the anti-B antibodies. Now, we're going to do positive and negative in just a moment. And I know I'm getting to the edge of the field of view here, but let me just make this clear. Before we even get into positive or negative, we need to talk about which blood types can you receive. If you were to get a blood transfusion, well, if I took someone's blood cells that had little A's on their surface from somebody else, could I put that into this person's plasma? And the answer is yes. You can always receive the same blood type because you cannot have antibodies against your own red blood cells. What if I took this person's blood cells and it had little B surface antigens and I put it into this person's blood? By the way, when you get a blood transfusion, we talked about this at the beginning of blood, they will fractionate your blood or they'll spin it and they'll collect all the plasma off, and then they resuspend the red blood cells, and you only get the blood cells. You don't get the antibodies that would be in their blood. But nonetheless, if I put this blood cell into this person's body, their antibodies would start to attack this and clump them all together. Their blood would clot and stop flowing, and it could kill them. Now, what if I put this red blood cell into this person's body? This antibody would still bind all the Bs. So they cannot receive anything that has a B on it if you have the anti-B antibodies. If I put this red blood cell in this person's body, they can receive it because there's no surface antigen that this antibody will clump or coagulate and stop blood flow. Now let's go to this person. Could I put this red blood cell with these surface antigens in their body? The answer is no. Their antibodies would bind it and kill them. So they cannot receive type A blood. As a matter of fact, they can't receive AB either. They can't receive anything that has an A, or their antibodies that are in their blood will clot those cells, clump them up, and kill them. They can receive type B blood, and they can receive type O because those antibodies won't recognize it. Let's go to AB. Can I put type A blood in this person's body? The answer is yes. Since they have A's on their surface, they can receive any cell that has an A on its surface because they don't have an antibody against that antigen. They can also receive type B blood because they have to lack the anti-B antibody. They can also receive AB blood, and they can receive O blood because there's no antibodies against the surface antigens. There's no surface antigens. So sometimes we talk about someone who's AB as being a universal recipient, particularly if somebody is AB positive. It's called a universal recipient because they can receive blood from anyone, A, B, AB or O, positive or negative, doesn't matter. And we haven't gone over positive and negative, we will, bear with me. So let's go to type O blood. Could someone with type O blood receive type A blood? The answer is no. They have anti-A antibodies which will clot that blood and kill them. Could they receive type B blood in a transfusion? And the answer is no, because they have anti-B antibodies, it will clot B blood. Can they receive AB blood? The answer is no. They have both antibodies to bind to both of those, and it will clot their blood and kill them. The only blood type they can receive is type O. Now notice, everybody can receive type O. And when you understand positive and negative, which we will in a second, you can say that type O negative is called a universal donor. They can donate to just about any person in the universe because it's not going to cause a clotting reaction. This is why blood banks are always starving for type O negative. It happens to also be the most common blood type, and I believe AB positive is one of the most rare, but nonetheless, you can read all those statistics. Okay? So let's talk about positive and negative so we can make sense of all of this. Let me see how much room I have on the camera and the space. Sorry for my unprofessionalism, but nonetheless, I got to do what I got to do. Okay? So I'm going to change the camera over just a little bit so we can talk about positive and negative. Now, positive, you can see my red stripe on the board, which tells me y'all are out of range of the video camera. Positive and negative refers to the presence of another antigen on the surface of your red blood cells. Okay. So, someone who's positive and someone who's negative. Let me see if that shows up on the screen. Again, I should do all this in advance and work it all out, but I don't. I just come in and do impromptu videos. So now, there's another antigen that can appear on red blood cells that is totally unlinked and unrelated to all of these. It is called the D surface antigen, also called the Rho antigen, or it's called RH. D, Rho, or the RH factor. They call it the D surface antigen, the Rho surface antigen, or RH factor. So, whichever one of these blood types that we are, I'm going to use type A just for an example. If I take a person who's type A blood, and I know they have the A surface antigens, and they also have the anti-B antibodies already in their blood. If they are positive, in addition to all of that, they're going to have another surface antigen called the D surface antigen, or also called the Rh factor, or Rho. So this person, if you're positive, you have the D or the Rh surface antigen. And you will have no anti-D antibody. You cannot make an antibody against your own red blood cells, or else you wouldn't survive in the womb. Now, if a person is negative, let's take the exact same blood type. Let's take someone who's type A. So this person would be A positive. This person's going to be A negative. If you're negative, it just means you don't have this D surface antigen. So the positive or negative tells you whether you have this in addition to whatever else you already have, or whether you don't have it. If I had a person that was B positive, I would have B's and D's. If I had someone who was AB positive, they would have A's, B's, and D's. If they're negative, no matter which they are, then they don't have the D surface antigen. But here's the kicker. This gene is not linked to the anti-D antibody. So usually this person makes no anti-D antibody. But there's a big deal. Unless they are exposed to positive blood. Because of how white blood cells and particularly lymphocytes work with specific immunity, if I took positive blood and I put it in this person's bloodstream, they will eventually develop antibodies from their white blood cells and clot that blood. So one of the things that we like to say is that positive can receive negative. Negative cannot receive positive. That's the negative in it. It can't receive positive blood. So anyone who's negative can receive their same blood type. If I'm A negative, I can receive A blood, but it has to be negative. If I put A positive blood into this person's bloodstream, over time they will develop antibodies, clot the blood, and kill them. This is why blood typing is so important. If a patient is losing blood, you need to replace that blood before they die, because it's delivering oxygen and nutrients, and the blood volume and blood pressure means good blood flow. If I stop blood flow to the heart or brain, my body shuts down and I die. So sometimes you need to give someone blood in surgery or after a massive injury like a gunshot wound or a car accident where they're bleeding profusely. But you better know the blood type and you better understand blood typing. You need to know who can receive what. So know this and know this. I will ask you some test questions on this lecture test, like a person with type A blood, someone who's A positive can receive which of the following blood types? And I might list 4 or 5 and you pick the ones they can receive. Or I might ask a question that says, a person that has AB blood has which of the following? And you would know that they have both the A and the B surface antigens and neither anti-A nor anti-B antibodies in their plasma. Okay? So know this information. If I were you, I would learn to draw this chart until you can teach it to a 10th grader. Well, if they can understand it, if you can stand at a board without your notes and teach it, then you're ready to take a test on it. Which is true of everything that we cover in this class. The only way I got good at this was sitting down and doing it until I couldn't stand it. When I was a college student, I used to go to the library, get a room with a marker board or a chalkboard, and I would be writing my notes and talking to myself. And I would have people stop and say, "Who are you talking to?" And I'd say, "I'm just learning the information. I'm learning to explain it so that I fully understand it." If you can teach it to someone else, you fully know it, so do that. Do it till you can't stand it, and then do it 3 to 5 more times, and do it every day until you take a test. And every time you master one thing, you start to learn a new topic. When you think you mastered that, go back to the beginning and see if you can do it all. And then learn the next lecture, and then go back and see if you can do all 3 lectures, sort of in a nutshell, in a synopsis for someone else. If you can teach this material, you know it. Anyway, thanks for watching. Oh, by the way, we got to do hemolytic disease of the newborn. I forgot to tell you this. So I'm going to erase part of the board. And then I'll shut up and you guys can start studying. If I have a mom who's pregnant with a baby, let's give her not quite a Kardashian figure, but nonetheless, okay. She's carrying a baby. Mom is negative. The baby is positive. If the umbilical cord or something breaks and bleeds and the baby's blood gets into mom, mom can start to make the anti-D antibodies. And if they cross the blood barrier there, they will coagulate the baby's blood and kill it. So a negative mom needs to, or anyone who gets pregnant needs to have the blood test. If you are negative and the baby is positive of any type, then you need to make sure you get a shot of a drug called Rogam. And Rogam will suppress the formation of anti-D antibodies so that the baby survives the pregnancy. Okay? Or else you'll develop antibodies against that. Once you do develop the antibodies, if you get pregnant a second time with a positive baby, it will not usually survive. So that can be a reason for many spontaneous abortions, the stoppage of the development of the embryo and the fetus. Anyway, I hope you had as much fun as I did. I hope you learned something. I hope you're enjoying this class and learning some really cool, interesting things about your body. I'll see you on the flip side. Thanks for watching.